So, good evening, guys. And today is the second lecture for the RNA biology class. And so, in the last lecture, what we studied about the RNA world hypothesis that was given by Booz, Carl Booz, Crick, and Orgel. Okay, and so that was given by Woos, Crick, and Orgel, and it states that RNA may have preceded proteins in functions. So, as we already discussed, that RNA has some catalytic functions. So that is why we are saying that it may have preceded proteins and catalyzed all reactions for survival, as well as it was the genetic material of some of the organisms, initial primitive organisms. So genetic material, that is why it might replicate on its own, and then which gave rise to the DNA. And then we discussed about different properties of the RNA. For example, it is a single stranded molecule and it has many secondary structures uh, because of the presence of the 2' OH group that is shown in this particular figure that we'll come upon. And then it has, as in some of the viruses, for example, tobacco mosaic virus and HIV, all these viruses have RNA as the genetic material, as well as it helps in the transmission of genetic information. For example, we studied that central dogma, which states that the information transmits from DNA, then to mRNA, followed by the protein. So, it is a major molecule that helps in transmission of information from DNA to the proteins. And then it helps in regulation of the gene expression. So, like we have microRNAs, siRNA, and different kinds of LNC RNA that helps in the regulation of gene expression during the need, and that the proteins that we don't need at particular time, so that the genes are switched off and the protein concentration goes down in the cell. So that is maintained by the different RNAs present in the cell types. Then for catalysis, for example, there is a peptide transferase, that is 23S rRNA, and 28S rRNA that helps in peptide bond formation, and then the spliceosome that is also composed of the RNA and the protein, combination of RNA and protein that catalyze different reactions. So it also has catalytic functions. Then cofactors for enzyme molecules, for example, the important molecule coenzyme A, that it also has an adenine moiety. So that comes from the RNA. Then energy storage in the form of ATP and different trinucleotide phosphates, that is ribonucleotide phosphates. Then the signal transduction, this comes for CMP, CGMP. These also proteins comes from the different ribonucleotides only. So these are the important properties as well as functions of the RNA that it performs in the living body. And coming back to the what we discussed about nucleotides, so RNA being a polynucleotide, so it is a polymer of nucleotides. So nucleotides has a nitrogenous base that is attached to a purine, pentose sugar, and it in terms of sugar, it has 2' OH group, that is a characteristic of the RNA, and that make it more reactive, which is attached to the 5' and 5' carbon is attached to the phosphate group. So this is the particular structure of the nucleotide. This all we discussed in the last lecture. So now moving forward, we'll discuss different types of RNA that we see in our body, and then we'll move on to the metabolism of nucleotides and how the ribonucleotides are majorly formed in the body. So this is the contents that we'll study today, and we are starting with the biochemistry portion of the RNA and how it is formed. Moving on, then we'll move on to the molecular biology concepts of the different RNA molecules. So, talking about the types of RNA, there are majorly on the basis of function that we see. So, basis of function, we divide the different RNAs. So one is the coding RNA and the other is the non-coding RNA. So the coding RNA that has the phenotypic information or the information that a gene gives to gene is transmitted to a protein that is done by the coding RNA. So the main type is the mRNA that basically forms protein. So this is the major type of coding RNA. Then comes the non-coding RNAs that may assist in transcription or as some regulatory functions. Okay, so sorry, assist in translation. This must be translation. So, different rRNAs and tRNAs helps in translation. So, this basically is like functional RNA rather than they are encoding some proteins, so they help in translation. And then there are regulatory, as we discussed, it also has some regulatory functions. So these are the regulatory RNAs that are present inside the body, like miRNA, siRNA, LNC RNAs that regulates the expression of a gene, and then SNU RNA, small nuclear and small nucleolar RNA, and then the gRNA that helps in gene editing. So we'll see a bit of the details of these RNA. And moving forward, so the first was the coding RNA, that is the mRNA, or the messenger RNA that we call about. So this is the only linear form of RNA that we see inside the body because it doesn't form any secondary structure. So and as we know that in case of proteins we call the linear form as the primary structure of the protein. So this is the primary structure of RNA. Then the major function is it carries genetic information copied from the DNA that is transferred to the protein. So for example, we have a gene encoding a protein. So, for the gene to come up with the phenotypic response, it forms a protein X. So, this gene to protein X is basically linked from gene that transmits information through mRNA that forms the protein. So this is basically the main function of the mRNA. And like we all know, there are two kinds of organisms. One is the prokaryotes and the other is the eukaryotes. So prokaryotes are basically the organisms that do not have a proper nucleus. So the genetic material is divided in the cytoplasm itself. So in the cytoplasm, everything is there that helps in the replication as well as formation of the DNA. And in eukaryotes, there is a proper defined nucleus that has proper chromosomes with histone proteins and different enzymes that are involved in the replication and all those things. So this is the basic difference between eukaryotes and prokaryotes. So that is why, because prokaryotes are small organisms, so to reduce the energy consumption, what happens in case of prokaryotes, there is a system of operons. So what are operons? So in prokaryotic system, the different proteins or different genes that have similar function that are arranged together. So the basic, very profound example of an operon is the lac operon that was uh, discovered by Jacob and Monod. Okay, so what happens? This is the particular structure of a prokaryotic mRNA, that is polycistronic. So what is a polycistronic means? So similarly, for example, we are stating operons. So in an operon, what happens? This is the starting point, and there are many genes: gene 1, gene 2, gene 3. Okay, then this is the termination point. So now, when this RNA will be formed, this will the RNA will also have a common promoter like this, and for gene 1, one RNA, gene 2, two RNA, and gene 3 followed by the termination codon. Then if different proteins need to be formed from these particular RNAs, then there will be a Shine-Dalgarno sequence that we'll talk about during the protein synthesis. So there will be a Shine-Dalgarno sequence that helps in the expression of this particular protein, and followed by this Shine-Dalgarno sequence for this particular protein, and the termination codon simultaneously for the previous protein. So, like this, there are multiple genes attached to each other in the form of a chain. So, this is one cistron, or the open reading frame. This is the other cistron that forms the protein part. So, there are multiple cistrons arranged in a series. That is why this RNA is called as polycistronic. Okay, now yeah, so as you can see in the diagram itself. Now, as we see in the eukaryotic mRNA, as the complexity increases from prokaryotes to eukaryotes, so what happens? We have more wide genome content as compared to the prokaryotes. So, in that case, there is more regulation in terms of production of particular proteins from DNA to RNA, and to RNA from RNA to proteins. So what happens in eukaryotic gene map? What we see, we have a replication or a promoter region that helps in particular production of the mRNA, followed by the coding sequence, or the ORF, that encodes a different protein, followed by the endpoint or the termination codon. Okay, oh sorry, the termination point of the mRNA. And when this particular mRNA go for translation, we have a 5' UTR, or the untranslated region, that helps in binding of the uh, ribosome to the particular mRNA, followed by the ORF, the main protein coding region, and the 3' UTR, or the untranslated region. And these two are, this is the 5' cap, and this is the 3' tail. So this basically is a property of the eukaryotes. Okay, so this is considered to be a monocistronic RNA. So single gene will produce only one RNA, followed by that forms the protein. Okay, so this is the major difference between the prokaryotic and the eukaryotic RNA. Okay, so any doubts till now? So if not, then we'll move on to the next point. Okay, so now moving on to the non-coding RNAs. So only coding RNA that we have is the mRNA, and the others are the non-coding RNAs, or the helper RNAs that we talk about. So then we have the tRNA. So tRNA is basically present during translation. So it helps during translation of the protein. So talking about its structure, then we'll go into its function. So it has a tertiary structure, the only RNA that has a tertiary structure, and that is L-shaped as it's shown here. Okay, and this is the secondary structure of the RNA, and it basically helps in the recognition of the codons. So the basic function that tRNA performs and how it performs we'll study in the protein synthesis or the translation process. But during this time, what we'll see that we have a mRNA chain that are consisting of the codons that are triplet base pairs. So tRNA has the role that it recognizes different codons and attach it to the amino acid. Okay, so this is the basic role of tRNA. Now, talking about its structure, moving back to its structure, so it has a 73 to 93 nucleotide long structure, and it is basically present in the cytosol because the translation process occurs in the cytosol, and various semi-autonomous organelles like mitochondria, chloroplast, etc. So it is present in these organelles also. Okay, because they have some amount of DNA present in them, so some of the proteins are formed from them, and it contains some of the tRNAs. And then talking about its structure, it has basically four loops. Okay, so one is the anticodon loop, this one that recognizes the codon on the mRNA. This is the mRNA chain, so it recognizes the triplet code and then takes the amino acid from different different amino acids. So different amino acids have different types of tRNA due to the wobble hypothesis because of the last base pair. Okay, now moving on to the different um, the second one is the T C loop. Okay, so psi is basically an unusual amino acid called as the pseudouridine. Okay, so this is pseudouridine, and this arm is basically the just 1 minute. So this T-C loop has a pseudouridine that is basically a structural change of uridine only. Okay, and this T-C loop hal functions as a help of peptidide transferase. So it helps in the function of peptidide transferase. Okay. Now we comes as the other loop or the DHU loop. Okay. So this D is called as the dihydrouridine. So there is a double bond between C5 and C6 of the uridine structure. That is why it is dihydrouridine. Okay, and it helps in proper fitting in the ribosome. Okay, now we have one of the arm called as the variable arm. So this variable arm is different in different tRNAs, and based on this, the length of the variable arm, there are two types of tRNAs, class 1 and class 2. Okay, so there are two kinds of tRNA. One class 1 has a shorter variable arm, and class 2 has a longer variable arm. And then the function of variable arm is basically like not known till now what can be the function of variable arm, but yeah, it has been seen that in different tRNAs the class 1 there the length is bit different. Okay, and then comes the acceptor arm that receives the amino acid, and it has a conserved sequence CCA that is present in all the tRNAs, and that is attached post translation transcriptionally. Okay, so this is not present when the transcription of this particular tRNA happens. So this is added at a like after the transcription has been happened or post transcriptionally, and these four bases are unpaired. Okay, that receives the particular amino acid. So this is the basic structure and the different functions of different arms of the tRNA. So this structure that we talked about is a clover leaf structure like this. Okay, and it has this is called as the secondary structure of tRNAs. Okay, and talking about different forms or different like types of tRNA in prokaryotes and eukaryotes. So prokaryotes have 30 to 45 different tRNAs based upon the how many amino acids they are need for a particular protein and different protein tropism in them. So based on that, they have 30 to 45 different tRNAs, and in eukaryotes we have 50 to 50, greater than 50 different tRNAs present inside a cell. Okay. So this was the first non-coding RNA, that is the tRNA or the transfer RNA, because it helps in transferring the information or help in the attachment of amino acids from a codon. Okay, so let's move on to the next RNA, that is the rRNA or the ribosomal RNA. So this RNA, as the name suggests, it is present in ribosome. So it is the RNA that is present in most number, or the highest amount of RNA present inside the cell is the rRNA. It is about 65% of the total RNA content, and it is associated with the proteins forming the ribosome. So it is a ribonucleoprotein. Okay, so this is the ribosomes, and they basically fold up to quaternary structure. As we saw in tRNA case, that the maximum level it can fold is the tertiary structure, that is the L-shaped structure that we saw, and this is the quaternary structure of the proteins. And with the proteins, for example, this is the rRNA molecule that it attached to the protein, forming a various kind of loop. That is the quaternary structure of the particular ribonuclear protein, and it has different modifications on the RNA. For example, the pseudouridine. These are the post-translation transcriptional modifications. So it may have pseudouridine, ribothymidyllic acid, as well as methylated bases. Okay, so these are the modifications that are present. Now, talking about the different ribosomes, as we have previously also discussed. So basically in prokaryotes there is 70s ribosomes. So 70s, and in eukaryotes it's 80s. Okay, so 70s means there is two subunits, the large and the small. Okay, and large subunit is called as the 50s and the small subunit is called as the 30s. The 50s subunit has two rRNAs, that is 5s rRNA and 23s rRNA, and the small subunit has 16s rRNA. Okay, so the 23S rRNA is basically a peptidyl transferase. So that helps in transferring the formation of the peptide bond inside the cell. 16S rRNA acts as a template for the RNA to attach between the two subunits of the ribosome. And 5S rRNA is basically the helping mechanism. It helps in the translation and proper positioning of the ribosome or across the mRNA. So these are basically the functions of the rRNAs present inside the prokaryotes. Now, when we comes to the eukaryotic ribosome, so it also has a large and a small subunit, that is 60S and a 40S subunit. And then in the 60S, we have three types of rRNAs, 5S that has a similar function, that it helps in translation. The 5.8 and 28S both combined forms the peptidyl transferase. Okay. So here we have only 23S. Here we have both combined combination of 5.8S and 28S, followed by 18S rRNA that helps in the positioning of the mRNA on the ribosomes, as well as it both 16S and 18S are evolutionarily conserved across the particular the domain or something. So they are also helped in like they also have been used in phylogeny to take out any of the functions or the evolutionary relationship between the organisms. So we study the 16S and 18S rRNAs. Okay, so this was the thing about the ribosomal RNA. Then comes the microRNA, the RNA that helps in the particular that helps in the regulation of the gene expression. Okay, so it is basically a 22 nucleotide long RNA, and it functional both transcriptionally as well as post transcriptionally. So the proper mechanism how miRNA functions and how RNA interference happens, that helps in the expression or the regulation of the expression of genes, that we'll talk later. And the major function or the property of microRNA is that it is not specific to a single gene, it targets multiple genes. Okay, so this is the major function of the microRNA. And so miRNA is basically is processed inside the nucleus. Okay, so it is formed and processed inside the nucleus. Okay, so this is the basic function of microRNA, and the basic details we'll study later during RNA interference and how the microRNAs are formed for different genes and how it protects against different microorganisms. We'll study later. So similar to microRNAs, we have the short uh, non-coding RNAs or siRNAs. Okay, so small interfering RNAs rather. Okay, so this is a bit larger, 20 to 25 nucleotide long, and then the function of siRNA is it targets the exogenous nucleic acids. Okay, so for example, there is a bacteria or the virus that infects our cells. Okay, it enters the cell and it releases its genome to get expressed, but there are certain RNA molecules that represses its expression. Okay, so this is the meaning of exogenous nucleic acid, that it represses the exogenous nucleic acid. Okay, so it is a form of the defense mechanism of the body. Okay, so then it is so specific for particular genes. Okay, like microRNA, microRNA targets many genes, but it this particular RNA is specific for specific genes. Okay, and then it maintains genomic integrity by silencing transcription from undesired loci. For example, if there is a promoter on the genome and there is a promoter and it forms gene, but we do not need this particular gene and there is another promoter here that forms the actual protein from that. So this particular siRNA will silence this particular gene that has no role, or we don't want that much longer protein. So the transcription will start from here only. Okay, this will form a functional protein that performs its particular phenotypic rescue or the phenotypic expression. Okay, and then we have different types of siRNA. For example, TASI, that is trans-acting siRNA, and it is majorly present in the land plants. Okay, depending upon the type or the function, we name these RNAs. Then we have the RASI RNA or the repeat associated siRNA, and this is basically formed during gametogenesis. So when the sperms and the ova are formed inside the body, so these particular RNAs helps in formation as well as different regulation of different genes required during gametogenesis only and not required during your adulthood. Then comes the pvRNA that also has the similar role, that is present only in the germ cells, not in other cells of the body. Then we have the scRNA or the scan siRNA. It is present in tetrahymena only and it helps in genome integrity of particular organism. Then we have the long siRNA or the LSI that is present in Arabidopsis and it is a defense mechanism against bacteria. Okay, so this is plant specific. So these are the types of siRNA that we talked about. Then there are smaller kinds of RNA present inside the body that we'll talk about. This is the SNR. So this is small nuclear ribonucleoprotein that basically helps in spliceosome or the splicing of the mRNA, forming different complex and all those things. So it basically helps in the formation of spliceosome and it is uridine rich. Okay, so the process of splicing, how it happens in the eukaryotic system and the spliceosome mechanism, we'll study in detail during the RNA processing. Okay, now moving on to the gRNA or the guide RNA. So basically, guide RNA helps in guiding the genome editing tools to a particular location. Okay, so for example, if we talk about CRISPR, so this particular guide RNA, if this is the protein, this is the gene that needs to be silenced or knocked out, so the guide RNA will guide it to the particular gene where the nucleus Cas9 can go and bind it. Okay, so this is the particular thing that we talk of, that we see in gene editing and how guide RNA helps in the gene editing process. So this is also another kind of RNA that we have talked about. Then we have the SNOT. Okay, so SNOTs are small nucleolar ribonucleoproteins that are coded by the nucleolus. Okay, so nucleolus also codes for some of the proteins. Basically, the ribosomes are formed in the nucleolus. So the nucleolus also encodes some of the RNAs called the SNOR that helps in the modifications or the new formation of modifications of rRNA and tRNA and the formation of ribosome. Okay, so this is the basic function of the SNOR RNAs. Okay, so these are small RNAs that helps in basically the control of the expression of different RNAs and all those things. Now, this completes the part of the types of RNA that are found inside the body. Okay, now moving on. So RNA, as we know that it has a, like DNA has a double helical structure. Okay, like this, it folds upon itself. So this is a helical structure, like this. RNA also forms a helical structure that is basically more stable as compared to DNA due to the presence of the 2' OH. Okay, now there are also different kinds of structures that are formed by the RNA. Like this is the hairpin loop that we generally see during the microRNA and siRNA production. Okay, then there is a bulge loop. What happens during this when the particular RNA also has a like similar sequence or a palindromic sequence in a short span after that. So it basically forms a secondary structure and folds upon itself. And where there is a non where there is a non-base pairing region, it folds upon itself and forms a bulge there. So this is how the bulge loop is formed. And then comes the interior loop, the similar kind of bulge loop, that for from the bulge loop, the interior loop can be formed where the bases are repelling to each other, and then the multi-branched loop. So after like folding upon itself, when the RNA has a large number or the large area that does not is not complementary to each other, but the other regions are complementary, forms the multi-branch loop. Okay, so these are the different secondary structures that the RNA is like the scientists have formed during their studies of RNAs that RNA forms. So, like now we'll be moving on to the biochemistry portion of the RNA, and this is all we have talked about till now. So now moving on to the nucleotide structure and how these nucleotides are formed. Okay, so talking about the nucleotides, so it has three basic components, that is a nitrogenous base, the pentose sugar, and the phosphate. So we'll start with the sugar first. Okay, this we have already discussed in the first class. Now I'm just starting with the clarification. Okay, just a bit basic description, then we'll move on to the process of synthesizing the ribonucleotides, the biochemistry component. Okay, so this the sugar is a pentose sugar, and the naming starts from 1, 2, 3, 4, and 5. So we write up in the form of a prime. So okay, and this is like in nucleic acids there are two kind of sugar. One is the ribose sugar that is present in the RNA or the ribonucleic acid, which has the 2' OH, and one is the deoxyribonucleic acid that contains the deoxyribose sugar, where the 2' OH is replaced by only the H. Okay, so this is the basic difference between the uh, RNA and the DNA. And here, one thing to note is the pentose sugar that we have is beta D. Okay, so this is the stereoisomerism that we'll be using. Okay, and all the sugars that are present in the living system are D. And then moving on, so the base is attached to the 1' carbon of the sugar, and the phosphate group is attached to the 5' carbon of the sugar. Okay, and yeah, so this is all about the sugar component of the nucleotides. When base combines with the sugar, it forms the nucleoside. So first, the base combines with the sugar, that is called as a nucleoside, and when the phosphate is combined to this particular nucleoside, this forms the nucleotide. Okay, so now talking about the bases, we have two kinds of bases present inside the body. First is the purines, and the other is the pyrimidines. Okay, So purines are two ring structure, and it has four nitrogens in them, starting from here, one from one position to the ninth position, and the nitrogen present is 1, 3, 7, and 9. Okay, and talking about the different kinds of purines, so we have adenine and guanosine. Oh, sorry, guanine. Okay, so in adenine there is a amino group present on the sixth carbon. So that's why sometimes it's called a 6 amino purine. And the guanine has a amino group at the second position, and an oxo group or the keto group at the sixth position. Okay, so these are the basic structural differences between the adenine and the guanine. Followed by the pyrimidines. Pyrimidine is a single ring structure, and it has two nitrogens at the one position and the three position. Okay, so it basically it has three kinds of we have three kinds of pyrimidines. So starting with the cytosine, it is present in both DNA and RNA. So these purines, adenine and guanine, are both present in the DNA and RNA. Okay, no difference between them. So cytosine has a amino group at the fourth position and the oxo group or the keto group at the second position. Talking about the thiamine, so it has three groups. Two are the oxo groups at the second and sixth position, and at the fifth position. No, sorry, second and fourth position, and at the fifth position we have a methyl group. In uracil, there is only one distinction between the thymine and uracil, that the methyl group at the fifth position is not present in the uracil. The other structure is same. At the two and fourth position, there is a keto group. So, talking about the presence, so uracil is only present in the RNA. Okay, and the so in RNA would contain cytosine as well as uracil, while DNA would have cytosine and thymine but not uracil. So this is the distinction between these two. And then coming about to the phosphate group that is attached to the C5 prime carbon of the ribose sugar. So there can be a single phosphate group, double phosphate group, or a triple phosphate group. Okay, so single phosphate group, as we see in the monophosphate or the AMP, that is adenosine monophosphate. Then the energy molecule that we need, the more phosphate is group. Phosphate groups are added to these particular compounds that stores more energy in terms of these bonds. Okay, so this is the ADP, that is diphosphate. And then comes the triphosphate, or the basic precursor for the synthesis of RNA or DNA during the transcription or replication process. So this is the triphosphate part, that is this particular is called as the adenosine triphosphate, that is an energy carrier. Okay, so these bonds that we have is a high energy bonds that stores a large amount of energy in them, and they release these energies by breaking the bond when needed during metabolism. Okay, so these are basically the structures that we see during the uh, uh, that are the parts of the nucleotides. Okay, now moving on to the nucleotide metabolism. So first we'll talk about the nucleotide synthesis. Okay, so we have basically two pathways for nucleotide synthesis. One is the Salvage pathway and the other is the de novo pathway. So de novo pathway is basically when you synthesize the whole uh, nucleotide from a single small molecules. Okay, for example, different amino acids, different groups, for example, formate and glutamine and all those things. Okay, so these molecules helps in the formation of nucleotides from scratch. So this is the de novo pathway. And Salvage pathway is basically when like during degradation, some of these nucleotides would go into degradation also. Okay, So when these nucleotides go into degradation, they tend to form like bases as compared to the complete nucleotide, and these bases can be transformed into nucleotide again. Okay, so this base that comes from the degradation of the nucleotide, we call that as a preformed base. Sorry. And Salvage pathway basically depends upon this preformed base and forms a nucleotide after that. Okay, so in the basic de novo, the site for the de novo pathway is the liver, and that too the cytosol. So it basically forms the triphosphates and sends it to the nucleus for its function. Okay, so it starts with the PRPP, that is 5 prime phosphoribosyl 1 pyrophosphate, or the precursor for the ribose 5 phosphate. Okay, so PRPP is basically the precursor for ribose 5 phosphate, and using various amino acids, ATP, and CO2, it forms a nucleotide. So this is the basic one step reaction for the de novo pathway for formation of nucleotides. Now, moving on to the salvage pathway, the basic site is plastids, where the recycling of the nucleotides occurs, and it forms the from the preformed bases. It is reconnected to the ribose unit. So PRPP, that is a source of ribose 5 phosphate, that is constant, and here the base is directly attached to this PRPP, and that forms the nucleotide. Okay, so this is the basic difference between the two salvage and the de novo pathway. So de novo pathway is the actual synthesis pathway, and salvage is basically like runs constantly with the uh, degradation part. Okay, so moving on. So first we'll discuss with the de novo pathway, then of both purines and pyrimidines. Then we'll move on to the salvage pathway and the degradation of the nucleotides. Okay, so moving on, first we'll do with the pyrimidine synthesis using de novo pathway. Okay, so as we know, pyrimidine has a single ring structure, and that too we have C, U, and T. So first C and U are formed, and then U basically is transformed to T. This is the step that is followed in the cell or the tissue or the living system that we talk about. Okay, so the pyrimidine molecule is basically when a base is attached to the ribose sugar. So when a pyrimidine molecule is attached to the phosphate group, okay, that is called as the orotate, and this orotate is further attached to the ribose 5 phosphate, followed by changing to different pyrimidines. Okay, so we have a process for C and U, and this further leads to the formation of T. And here the bases are not intermediates, and it is arranged over the ribose 5 phosphate. Okay, so here we can see that the aspartate. So this is a basic pyrimidine ring that we saw in the last slide also. So out of these four from 1, 4, 5, and 6. So this nitrogen and these 4, 5, 6 carbon is comes from the aspartate molecule. Okay, and this second and third, the second carbon and the nitrogen, this comes from the carbamoyl phosphate. Okay, so what happens? The formation of carbamoyl phosphate we'll talk about. So basically, what happens? The glutamine that has a amide side chain. Okay, reacts with the bicarbonate ion. Sorry, it's minus. So the bicarbonate ion that forms the carbamoyl phosphate. This is formed by the carbamoyl phosphate synthetase type 2 enzyme. Okay, so this also requires ATP, and it is basically maintained in the cytosol, this reaction, and this particular enzyme is present in the cytosol. There is another carbamoyl phosphate that forms during urea cycle, that is not, that we are not talking about here. So here the glutamine and bicarbonate forms the carbamoyl phosphate. This carbamoyl phosphate acts as a precursor for the pyrimidine synthesis de novo. Okay, so that we'll talk about in detail in the coming slides. So, talking about why we discussed this first, because such type of questions are asked in competitive exams. For example, there can be a question that asks that the fourth and fifth carbon of the pyrimidine chain are is formed from which of these amino acids. Okay, so the correct answer would be aspartate, and the confusing question could be that the third nitrogen would come from which of these amino acids. So this will be glutamine amino acid. Okay, or there can be carbamyl phosphate also. So carbamyl phosphate forms these two particular atoms, that is the C2 carbon and the N3 nitrogen, and the nitrogen first, fourth, fifth, sixth carbon forms from the aspartate. Okay, so now moving on to the major process of the de novo pathway of nucleotide synthesis. So like we talked about, there are two molecules basically involved, the aspartate and the carbamyl phosphate. Okay, so first what happens, the aspartate molecules is attached to the carbamyl phosphate, releasing a monophosphate. Okay, the PI, inorganic phosphate, is released, and here forms the N carbamyl aspartate. Okay, why N? Because this carbamyl moiety is attached to the N group. Okay, for example here. Okay, so yes, so this moiety is attached to this particular N. So that is why N carbamyl aspartate. This is the first intermediate in this step. Okay, now this particular reaction is being done by the aspartate transcarbamylase. So here what happens, the carbamyl is transferred to the aspartate molecule. That is why this is trans, because this group is transferred to the aspartate group. Okay, now this particular N-carbamyl aspartate moiety, this we are seeing here, okay, is transferred to L-dihydroorotate. That is the basic pyrimidine structure that we see. So this is a pre-hand or the precursor of the pyrimidine molecule called as the orotate. Okay, this is also being asked in various competitive examination, that what can be the precursor of pyrimidines. So this is orotate. Okay, so here the first compound, the second compound, that is the L-dihydroorotate, is formed using the enzyme dihydroorotase. Because in the primitive era, we what how we name the enzyme from the product that it is forming plus the type of reaction and then the ase. Okay, so here dihydroorotate is forming, so the enzyme name is called as the dihydroorotase. Okay, now this the third reaction is a dehydrogenase reaction. Okay, redox reaction rather. So this L dihydroorotate forms the orotate. Okay, so what happens in this case? The double bond is formed between the second and the third carbon. Okay, so that is why it forms the orotate with the help of NADH, that is the main redox converter inside the bond. Okay, so here one molecule of NADH is formed. So here the orotate is formed. Then this particular orotate basically is converted to the oro- orotidylate. Okay, so what happens? This primitive pyrimidine molecules is attached to the ribose 5 phosphate in the form of PRPP. So this is 5 prime phospho ribosyl pyrophosphate. 1 pyrophosphate. Okay, so this is a precursor for the ribose 5 phosphate. So as we can see here, so orate is attached to the ribose 5 phosphate. So this is the orate molecule that is attached to the 1 prime carbon of the ribose sugar, that is the ribose 5 phosphate releasing pyrophosphate group. Okay, that is attached to the 1 prime end of the particular sugar, forming orotidylate. Okay, so this is called as the orotate monophosphate. This is the first monophosphate that is formed during pyrimidine biosynthesis. So this is orotate monophosphate. Okay, so there the enzyme involved is orotate phosphoribosyl transferase. Okay, now this orotidylate, this is the precursor for the pyrimidine monophosphate. Now this is changed to uridine, or we have to change it to UMP as well as CMP, or the uridine prime pyrimidine and cytosine. Okay, so now this particular orotidylate is converted to uridylate or the uridine monophosphate. Okay, so now this is catalyzed by the enzyme orotidylate decarboxylase. So what it does is, because in uridine or the UMP we do not have a methyl group. Okay, so here we have the double bond oxygen, double bond oxygen. So we have to remove this particular C double O bond. So carboxyl bond we have to release. So here this carbon dioxide is released and this forms the uridine monophosphate with the help of the enzyme orotidylate decarboxylase releasing carbon dioxide, one molecule of carbon dioxide. Okay, so now we forms the uridine monophosphate. This is uracil. Okay, now this UMP needs to be converted to UTP or the uridine triphosphate. So this particular thing is done by the kinases, as we all know, because this will be done with the help of ATP. So now ATP comes, two molecules of ATP comes, it transfers its one phosphate, sorry, two phosphates from this two molecules of ATP to the this 5 prime phosphate forming UTP. Okay, here two phosphates are arranged. So why we are using 2 ATP instead of a one ATP molecule? Because if we use one ATP molecule, that will form AMP. Okay, and this AMP will be needing uh, two molecules more to convert it to ATP. So here, instead of saying that we needed one molecule of ATP that is converts to AMP, we use two molecules of ATP that forms two molecules of ATP. Okay, so this is the first reaction that we are talking about. Sorry, this is the reaction of forming the UTP. Now, when we convert UTP to the CTP or the cytidine 5 prime triphosphate, what happens? The glutamine comes into play, and this particular NH2 group of the cytosine is provided by the glutamine amino acid, and here one molecule of ATP is required to form the cytidine triphosphate. Okay, here the oxo group is converted to amino group. That is because of the glutamine reaction. Okay, yeah. So starting, we have aspartate plus carbamoyl phosphate. Okay, this indirectly forms orotate. So this forms orotate, and orotate forms the orotate monophosphate or the orodilate, which forms UMP. UMP forms UTP, and UTP forms CTP. Okay, so you need to remember the sequence that first UMP or the monophosphates are formed, and then it converts into triphosphates. Okay, because such kind of questions are asked in competitive examinations, that what kind of phosphate is formed during the reaction. Okay, and then first the UMP is formed, that is uracil, and that is converted to cytosine. Okay, so uracil is formed basically from the orodilate decarboxylase, the major enzyme forming UMP, and cytidylate synthase or synthetase is helpful in formation of cytidine 5 prime triphosphate or the CTP. That is the major pyrimidine or the cytosine nucleotide. Okay, so this was the process of formation of the urine and cytosine nucleotides. So, talking about the number of reactions that were required, so these were three steps, and till UTP it was three steps. So, total six steps were required for UTP formation, and seven steps were required for CTP formation. Okay, so sometimes was also the question, that how many steps are required for formation of the CTP from the aspartate residue. Okay, so this is the basic de novo pathway for pyrimidine synthesis for CTP and UTP synthesis. Now we'll go on to the thymine triphosphate synthesis. Okay, so what happens when we have UMP? Okay, with the help of ATP it forms UDP, and then because thymine is only present in the DNA form, so first what we have to form the deoxy UDP. Okay, so this ribonucleotide reductase helps in the formation of DUDP with the help of the NADPH. Now, this DUDP or the deoxy UDP is converted to the deoxy UMP or the monophosphate group. Okay, and then what comes, the thymidylate synthase that helps in the through tetrahydrofolate or the methylene tetrahydrofolate. This helps in the addition of the methyl group to the to the uracil moiety and forming dihydrofolate, forming thymidine monophosphate. Okay, so this was the small reaction that helps in the formation of thymidylate or thymidylate monophosphate. Okay, with the help of the enzyme thymidylate synthase. Now, this particular reaction is of particular interest during chemotherapy. Okay, so what happens when this particular TMP will not be formed? Then the cell will die or move on to the apoptosis. Okay, so what happens? Scientists have discovered different drugs so that they can inhibit this particular reaction of thymidylate synthase, So that will not form thymine or the TMP that will lead to the apoptosis of the particular cell type. So there are two drugs called as methotrexate and aminopterin. Okay, they basically inhibit dihydrofolate reductase. So what will happen? This dihydrofolate needs to be changed to methylene tetrahydrofolate. Okay, so if this reduction is not happening, okay, so this amount will go down. So the thymine synthesis cannot happen without this because methyl group will not be transferred. So here the enzyme is dihydrofolate reductase. So here these particular drugs, aminopterin and methotrexate, inhibit this particular enzyme that leads to the decrease in quantity of this methylene tetrahydrofolate that leads to decrease in TMP production, leads to apoptosis of the cancer cells. So this is the particular way how scientists discovered that okay, this particular therapy can be useful for uh, like lowering down the cancer cells inside the body. Okay, so here comes the end of the pathway for the pyrimidine synthesis using de novo pathway. Now we'll talk about some of the regulations of this particular pathway and how the feedback loop is being performed. So first, the feedback loop is basically, let's see in this reaction itself. So as we see that CTP is the direct regulator of the aspartate transcarbamylase. So this is a feedback inhibition loop. So as the CTP amount increases, this leads to the inhibition of this particular enzyme. Okay, so that it will not act on aspartate and will not proceed with the de novo synthesis. Okay, so when CTP is found formed in adequate amount, so this thing happens. So this is the major feedback loop. Okay, so now the other thing is that the ATP is basically the reversal of this feedback loop. Okay, so when the CTP is not there, so this is the normal activity of the enzyme when aspartate is present. Okay, but when CTP is there, this leads to decrease in the activity of this particular enzyme. Okay, so this is the Km. So when the CTP is not there, so the Km is high. Sorry, Km is low. And when the CTP is there, or there is a feedback inhibition happening of that particular enzyme, then the K point 0.5, that is the inhibition constant, is high. Okay, now what happens? There is a mechanism that the scientists have seen that if ATP concentration is also high, in this case when the CTP concentration is high, that reverses the inhibition. Okay, leading to the decrease in the inhibiting inhibitor constant. Okay, so this is a phenomena that has been discovered till now. When ATP is there, this leads to the reversal of the inhibition. Okay, so this is basically the feedback mechanism of the pyrimidine synthesis that happens inside the body. So now moving on to the purine biosynthesis pathway. Okay, so it's basically a particular difference between different molecules. So, like we also seen in pyrimidine synthesis that on ribose 5 phosphate, the pyrimidine molecule first will be arranged and then it will be formed. So similar thing happens here also. The purine molecule is assembled over the ribose 5 phosphate, followed by changing it to different purine molecules. And bases here are also not intermediates. Okay, because the bases are formed on the ribose phosphate. So now first we'll see that what atom is formed from which amino acid. Okay, so the basic ring of pyrimidines, as you remember, was formed from aspartate, glutamine, and bicarbonate ion. Okay, these two forms carboxyl phosphate. So basically these three helps in the formation of the ring of pyrimids. Now talking about the purines, there are different molecules that helps in different synthesis. So starting from here, this is the first carbon, first nitrogen, sorry. Okay, so the first and the ninth nitrogen comes from the glutamine amide. Okay, the second carbon and the eighth carbon comes from formate. The third nitrogen comes from the aspartate molecule. Fourth carbon comes from the carbon dioxide. And fifth and sixth carbon and seventh nitrogen comes from the glycine amino acid. Okay, so this is how the purine molecules are arranged over the ribose 5 phosphate. So here what happens, I'll repeat once more. The first and the ninth nitrogen are comes from the amide end of the glutamine molecule. The second carbon and the eighth carbon comes from the formate molecule. The third nitrogen comes from the aspartate molecule. The fourth carbon comes from the carbon dioxide molecule. The fifth and sixth carbon and seventh nitrogen comes from the glycine molecule. So three amino acids involved: aspartate, glutamine, and glycine. Formate and carbon dioxide provides different carbon atoms. Okay, so this is the different molecules that provides different carbon atoms during the purine biosynthesis. Okay, so now moving on to the process that is involved during purine biosynthesis. It's a bit complicated one, but we'll see how the things work. Okay, so the starting is the PRPP. In the pyrimidine biosynthesis, the starting was aspartate and carbamoyl phosphate. Here, the starting is PRPP and glutamine. Okay, so now this is the PRPP molecule that provides ribose 5 phosphate. So here the 5 PRPP molecule is attacked or the is joined with the glutamine molecule. Here it forms a nitrogen molecule that is the first carbon, first nitrogen that I can see. Okay, that we'll see later what number of nitrogen that it will form. So the amide group nitrogen is attached to the phosphate, ribose 5 phosphate moiety, forming 5 phospho beta D ribosamine, because this is the amino group. So it will it will be named as amine of this particular moiety. Okay, so this is the first carbon of the sugar. Now, this particular reaction is governed by glutamine PRPP amidotransferase. Why amido? You have to answer in comment. Okay, why this is called as amido group? Okay, now this particular amino group has been attached to the ribose 5 phosphate. Now comes the second amino acid, that is glycine, that uses ATP molecule. Okay, so this glycine forms the fifth, sixth, and seventh atom of the purine ring. So this attached to the ribonucleotide. So this particular NH2 attaches these three: CO, CH2, and NH3. Okay, so as we know the structure of glycine, we have CH2. Okay, so here OH is released, this particular OH is released, and the other moiety is attached to this particular NH, and here from here H is released. So a molecule of water is also released here. Okay, with the help of ATP. So here the name of the enzyme is GAR, or glycinamide ribonucleotide synthetase. Okay, and this particular molecule that is formed is called as glycinamide ribonucleotide, because the most of the glycine moiety is attached to this particular NH group on the ribose 5 phosphate. Okay, so this is the second intermediate formed during the reaction. Now, this particular intermediate is attached to the folate group. So now comes the carbon addition to the ring. So now the N10 formyl tetrahydrofolate basically attaches a carbon to the ring, forming formyl glycinamide ribonucleotide, or the FGR. So here the carbon of this particular group is attached to the ring, purine ring. Okay, so here the because the formyl group is being transferred, formyl group is basically C, uh, carbon attached to the, this is the formyl group. So this is being transferred from the formate to this particular ring. Okay, so here the reaction will be GAR transformylase. Okay, so here the formyl glycinamide ribonucleotide is formed. Now, this particular FGR is again attacked by glutamine, because as you remember, there is another nitrogen atom that is being attached to the this particular ring. So the second ring formation is started here. Okay, so this is the ninth nitrogen that attaches to the ring, and here also the one molecule of ATP is formed, is used for the reaction. Okay, this particular compound that is formed is called as formyl glycinamide ribonucleotide, or the FGAM. Okay, glycinamidine. Here it was glycinamide. Here it is glycinamidine, because the double bond nitrogen is being attached here. Okay, so this is governed by the enzyme called as FGAR amidotransferase. Okay, so now one molecule of glycine has donated its group, formate has donated its group, and two molecules of glutamine has donated its group. Now, what happens? The five-membered ring of carbon is formed. Okay, sorry, this is the first carbon. This is the sorry, first nitrogen, this is the ninth nitrogen. Okay, so now what happens, this five membered ring is completely formed in the next reaction using ATP. So what happens, this is the imidazole kind of structure. So this is 5 amino imidazole ribonucleotide or the air. Okay, so this 5 membered ring is completely formed here. Okay, so here ATP is used, and because the bond is formed between these two, the water is also released. And this is the enzyme involved is FGAM cyclase. Okay, so just a second. So this is FGAM cyclase, where this particular five membered ring is being formed completely. Okay, now moving on to the next step. What happens? This air, the imidazole ribonucleotide, is particularly formed the bicarbonate ion and the ATP attached to the second ring. Okay, forming the N5 carboxy amino imidazole ribonucleotide. Okay, so here the fifth carbon. Okay, is formed here, and then what happens? Huh. There is so this particular compound is formed, and the enzyme involved is N5 CAIR synthetase. Now, due to rearrangement of the next step is the rearrangement of this particular compound to this. So, what how this happens with the help of the enzyme carboxylase. Okay, or N5 CAIR mutase. So this particular air can go directly add CO2 and through this position through carboxylase enzyme, or it can add through go through this particular path and use the bicarbonate ion, and through two step process it will form this kind of structure, carboxy amino imidazole ribonucleotide. Okay, now comes aspartate into picture. Okay, so this aspartate uses a ATP molecule and attaches itself to this particular carbon of the ring, and which forms N-succinyl 5-amino imidazole 4 carboxyamide ribonucleotide. So the short form is SAICAR, and this particular enzyme name is SAICAR synthetase. Okay, so now the aspartate molecule also has donated its particular body to the ring structure. Now, this particular ring is attached to the fumarate. The fumarate molecule is being released from the particular aspartate molecule, and only the amino group is there. And this particular compound is called 5 amino imidazole 4 carboxyamide ribonucleotide or AICR. Okay, and this is basically a rearrangement reaction where the fumarate molecule is being released from the molecule. So the enzyme name is called as cyclase. Okay, now the formate group, another carbon from the formate group is being attached, so that attaches to the nitrogen molecule, forming N-formyl amino imidazole 4-carboxyamide ribonucleotide, or FAICR, where the this is also a transformylation reaction. So the enzyme name is AICAR transformylase. Okay, so this particular ring, the second six-membered ring, is also formed. The left only the left out is the bond formation between these two molecules, or these two moieties. So now this water is released, and the first precursor for the purines is formed, that is inosine monophosphate. Okay, so this molecule is called as inosine. Okay, so this is how the rings or different atoms are arranged in the moiety. So first, the five-membered ring is formed using glutamine amide bond, glycine, followed by the formate group, followed by the ring formation. Then comes the glutamine, carbon dioxide, aspartate, and then the last step is the formate. So formate provides two carbons, glutamine provides two nitrogens. Okay, glycine provides two carbons and one nitrogen, and aspartate provides one nitrogen, and carbon dioxide or carbon bicarbonate ions provides one carbon. This is how the purine ring, the basic ring structure of purine is formed on the ribose 5 phosphate, or the PRPP, that is the precursor. So this forms the inosinate or the inosine monophosphate. Okay, now this particular inosine monophosphate is converted to GMP or AMP, that is guanylate or adenylate, that we'll see in the next slide. So this inosine follows two paths. First for the formation of adenylate, and second for the formation of guanylate. So first we'll see for the adenylate part. So this IMP structure, first it is attacked by the aspartate and the GTP molecule as an energy precursor. So this attacked aspartate moiety is arranged as we have seen previously also. Then the fumarate molecule is released, leaving this particular NH2 attached to the ring structure, and at the sixth position, sixth carbon. And here the rest of the fumarate or the rest of the moiety is released, just leaving behind the amino group. Okay, so this particular moiety is adenosine monophosphate, or the AMP, yeah, adenylate. So here two enzymes are involved: adenosuccinate synthetase and adenyl succinate lyase. Okay, now comes the guanylate formation. So here what happens? One molecules of water and the redox reaction happens using the enzyme IMP dehydrogenase, and releasing one molecule of NADH, and here one oxo group or keto group is arranged. Okay, this is xanthylate or xanthine monophosphate. Okay, now what happens? One molecule of glutamine releases its amino group from the amide group, and using energy in the form of ATP, it forms the NH2 group in the second position. Okay, this forms guanylate or guanosine monophosphate. So in case of ATP, aspartate releases its amino group, and in case of GTP or GMP, the glutamine molecule is being released. Okay, so this is the formation of different purine moieties from IMP. Now moving on to the feedback mechanism of the purine biosynthesis using de novo pathway. So what happens, this is the basic feedback loop that follows. And what happens first is the IMP. Okay, when IMP is more in amount, so what happens, this basically inhibits glutamine PRPP amidotransferase, which leads to formation of 5 phosphoribosyl amine. That is the first or the second step. Okay, now this IMP leads to the formation of AMP and GMP. Now, as you noticed in the previous slide, that here for the formation of AMP, GTP is required, and for the formation of GTP, ATP is required. Okay, so this is a cross reaction. So here, when the amount of AMP is high, so what happens? It basically inhibits succinate synthetase, that is a feedback inhibition loop, and this further inhibits this particular enzyme also through allosteric inhibition. And in GMP, when GMP is high in concentration, it inhibits this particular enzyme that leads to the reduction in AMP formation and as well as this particular enzyme. Okay, so this is how the feedback mechanism of purine biosynthesis works. And GMP also acts on IMP dehydrogenase as an allosteric inhibitor so that it will decrease its activity during the process. So this is how the AMP, GMP and IMP follows the feedback loop for the reduction in synthesis of these particular intermediates. Okay, so this leads to the end of today's lecture, and we'll meet tomorrow with the continuation of the metabolism of nucleotides, including Salvage pathway as well as the degradation of the nucleotides. And we'll also discuss some of the previous year questions from different competitive examinations. Okay, so if you have any doubts, you can write in the comment section. And thank you for today.